Hello, everyone. Welcome to CyVerse Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar is on workflow management using Snakemake, presented by Sateesh Peri. Sateesh is a doctoral student in genetics here at the University of Arizona in Dr. Eric Lyon's lab, also an instructor for both the CyVerse Foundational Open Science Skills and Container Camp trainings, as well as for the data and software carpentries. For those unfamiliar with CyVerse, we are an NSF-funded cyber infrastructure project, and this free webinar series is designed to fulfill a key part of our mission to train scientists like you on how to use CyVerse's computational resources. First, let's quickly take care of some housekeeping, and then we'll start the webinar. Sateesh's presentation today is roughly 30 minutes with time for question and answers at the end. Please open the Zoom chat window and type any questions there for Sateesh. His slides and the webinar video recording will be posted on our website webinar page later today for you to review at any time. Even before the COVID-19 pandemic radically changed our ability to travel, CyVerse was working on tweaking the curricula for both Container Camp and for our Foundational Open Science Skills Camp to accommodate remote learning and to reach a larger audience. So please watch our website for information on those trainings. And without further ado, I am pleased to introduce Sateesh, who is CyVerse's favorite snake charmer. Hi, Sateesh.
Thank you, Tina. Thank you so much. Hello, everyone. I hope all of you are doing safe out there. So today's webinar topic would be workflow management using Snakemake. And I am going to paste the link to read the docs for today's tutorial in the group chat. So let's begin by first launching our app, which contains Snakemake. And while it's launching, we can go over the introduction. So if you see the read the docs page today and scroll down, there are two ways you can launch the Snakemake VICE Jupyter Lab app. That is a Jupyter notebook which has Snakemake and other pre-installed in it. For first-time users or just for a quick launch, by right-clicking the Powered by CyVerse button and opening it in a new tab, it'll directly take you to the Snakemake VICE app within the discovery environment. So to log in, please enter your CyVerse credentials, and you will be redirected to the discovery environment. And within the discovery environment, if you have used the quick launch button, it will directly open the Snakemake VICE app for you. But if you can also find this app from within the discovery environment. To do that, click on the apps tab within the discovery environment, where it will bring up the apps window. And there you have to search for Snakemake, and it should pop up the Snakemake VICE app. Just give that a second. There we go. So if you see, this is the Snakemake VICE app that is available within the discovery environment. So you can click on the app, and either you can name your analysis or leave it defaults and click on launch analysis. And this will now submit a job to the discovery environment with a request to launch a Jupyter notebook. And once you have clicked on launch analysis in the notifications tab, you can see that the job has been submitted and it's currently running. And just right next to it, you can see a link that says access your running analysis here. So if you click that, it'll open a new tab within your browser, and it'll redirect you to a Jupyter notebook. So I will now give a minute or two for people to launch their Jupyter notebooks, while in the meanwhile we go over the introduction for Snakemake. for the introduction, I'm going to use the PDF that I have listed in the Read the Docs. Okay, so as we wait for our Jupyter Notebooks to launch, the main motive of today's webinar is Snakemake. The premise is as projects grow or age, it becomes increasingly difficult to keep track of all the parts and how they fit together. So Snakemake is a workflow management system that aims to reduce the complexity of the workflows by by providing a fast and comfortable execution environment together with clean and modern specification language in Python style. Uh, and this is a nice image that reflects some of the complexities as your research project goes. So on the x-axis, we have reproducibility, and on the y-axis, we have scalability. And what you see over here is the journey of data from raw data to processed data, which you use in publications. There are a series of transformations and intermediate steps that occur. And for the sake of reproducibility, it is as important to record each step and each stage of transformation for these data. And a workflow management system is where a tool like Snakemake comes in, which helps you manage and record all the steps that happen within your data analysis. So with that, I am actually gonna give my acknowledgements right now, not to the end. Special thanks to instructors at NBIS, National Bioinformatics Institute at Sweden, for the tutorial that we are going to use in this webinar. This tutorial is part of a 6-part course on tools for reproducible research. You can read more about the course over there. This course was actually supposed to happen next month, but it's been canceled due to the current pandemic. But I'm grateful for the instructors for making the slides available here. All right, so I hope your Jupyter notebooks have launched within the CyVerse Discovery environment. So what you see here is a Jupyter notebook, and we will be using the terminal access from within the Jupyter notebook and also the text editor for editing our workflow. All right, so now assuming that you have your Jupyter notebook launched, the link to the tutorial is in point 7 of the Read the Docs. So go ahead and open that in a new tab. So just for the sake of real estate on your screen, you want to have your Jupyter Note Lab on one tab and the tutorial on the other for the ease. Okay, so this is the tutorial for Snakemake. You can follow along with me as we go. Or I just want to also state out that this is a self-paced tutorial. You can follow this even at your own pace after this webinar for more detailed documentation. So we will skip the environment creation part because this particular Jupyter notebook has all the tools required installed already, and we'll directly go to the practical exercise. Okay, so to start off with Snakemake, in your Jupyter Lab, open a new terminal, and I'm going to increase the font here. Okay, I hope you can see that. Okay, so to begin with the basics, we'll create a very simple workflow from scratch in order to show the fundamentals of how Snakemake works. The workflow, which we will take, has 2 files, input a.txt and b.txt. And the purpose of the workflow is to convert the text within these files from lowercase to uppercase, and then to concatenate both these files into one file. So to begin with, run the following shell commands. It's totally okay to copy paste from the tutorial. The first one will make an empty file called Snakefile. That's what the touch command uses. And the next two commands will generate two files containing some arbitrary text. So I'm going to copy the commands and paste it into my terminal within the Jupyter notebook. And make sure you press enter at the last command. And you can see that we have created in your Jupyter notebook, towards your left is the file explorer, and there's a refresh button on the top of the file explorer. You can refresh and you can see that we have created three files, a snake file and two text files, where we have added, you can open the text file and you can see that we have added text in lowercase to both the texts. So now let's open our snake file, which is nothing but a text file in the Jupyter notebook. Now, a Snakemake workflow is based on rules which take files as input, perform some type of operation on it, and generate some files as outputs. Here is a very simple rule that takes a.txt as input, produces a.upper.txt. So let me paste the rule into my snake file. So as I was saying, here is the rule. A rule has a name. Every rule has a name. And you can see the rule here. The name of the rule here is convert to uppercase. You can name the rules however you want, but please make an effort to name your rules in a way that it makes it easy to understand the purpose of the rule, as rule names are the one of the main ways you interact with your workflow. Okay, so you can see that in this rule, we have clearly demarcated the input, the output, and the shell command that needs to be run. So you can see the shell section here contains the shell commands that convert the lowercase letters into uppercase and send it to the output file. And if you notice, here we are referring to the sub elements within the rule using curly braces. So these input and output in curly braces refer to the sub elements within the rule. So if you're not familiar with Bash, this tr command that you see over here is basically taking the input within a.txt and converting everything within lowercase to uppercase and sending it to the output file, which is a.upper.txt. So here is a simple rule. Now let's save a rule by pressing Control S. And now that I've saved my snake file, go back to your terminal, and now we'll try to run our first Snakemake workflow. So now here is an important point to understand. When a workflow is executed, Snakemake tries to generate a set of target files. Now, target files can be specified via the command line, such as by saying snakemake make a.upper.txt. Because you can see in this particular rule, this is the output that we are expecting. This is the target that we want Snakemake to make. So that is a.upper.txt. So now you can say in your terminal, when you say type snakemake, make a.upper.txt. That is the target file that we want Snakemake to make. And before we run the Snakemake workflow, I'm going to introduce three different flags for Snakemake execution. They are -n, -p, and -r. Now, -n is a very handy feature within Snakemake. It's called the dry run feature, where it'll show what Snakemake plans to do without actually running or executing anything. Now, the -p flag stands for printing the shell commands that Snakemake will execute. And the -r flag is the reason for running a specific rule. You might not, the logic behind the -r, the reason for printing the reason might not be clear right now, but hopefully it will by the end of the tutorial. So now when I press enter, and when I ask Snakemake to generate a.upper.txt, you should see some output like this that says it's building the DAG of jobs. And you can see the jobs, it's saying that there's one job to be done. And this is the shell command that it will run in order to get the target file, which is a.upper.txt. So, but remember, we use the -n flag, which is the dry run. So this is what Snakemake is planning to do, but it has not done yet. So now, in order to actually execute the workflow, remove the -n flag. By pressing up, you bring up the previous command, and remove the -n flag and press enter. And you can see that and if you refresh your file explorer in the Jupyter notebook, you can see that a.upper.txt has been created. And you can see that the lowercase uh, text within a.txt has been converted to uppercase. So great, our Snakemake rule works. So you can see that, okay, so now what happens if we try to run Snakemake again? And so try bringing up the previous command and press enter. And you can see now that Snakemake says nothing to be done. So now this is a feature. So it turns out that Snakemake only reruns jobs if one of the input files is newer than the output files, or if one of the input files will be updated by another job. Or in a simple sense, Snakemake is keeping track of the timestamps of the files involved in the workflow. And only when the timestamps are updated will the Snakemake realize that, oh, this is a new file and it has to rerun a rule again. So this is how Snakemake keeps track of everything in the workflow is up to date. So, okay, so in our previous rule, we have asked Snakemake to generate a.upper.txt. But what happens now if we ask Snakemake to generate b.upper.txt? So I'll go ahead and ask Snakemake to make b.upper.txt. But I'll use the dry run flag just to make sure that Snakemake tells me what it's planning to do, and also the p and r flags. And you can see when I ask Snakemake to make b.upper.txt, there's a missing rule exception. And if you go back to your Snakefile, that is true, because there is, because of how we wrote this rule previously, where we have explicitly stated the name of the file into the rule, there is no rule to make b.upper.txt, right? So now we can either copy this rule and make a separate rule, a new rule just for b.upper.txt, but it's just going to become cumbersome, and it's not intuitive. So here is where one of the most important features of Snakemake comes into play, which is named wildcards. So rather than how we wrote the rule earlier, where we said a.txt, we can change the name to a named wildcard. And in this case, a wildcard is anything which is between curly braces. And you can name your wildcard anything that you want. So in this case, if you replace a.txt to some name in curly braces .txt, and again here in curly braces, some name .upper.txt, in this way, now we have generalized the rule. And because when we ask Snakemake to make a target file, Snakemake will replace the wildcards with the value of the target of the input files and also generate those. So what happens here is that let's actually run this, save the file, and let's try to run b.upper.txt now with the -n flag. And now you can see that the wildcards in the Snakemake rule have been replaced by what we specify in our target file execution, what we specify on the command line. And it now will generate the rule and run the job for you. So what happens here is that Snakemake looks at the rules it has available, and tries to assign values to all wildcards so that targeted files can be generated. In this case, it was quite simple. You can see that, that it says wildcard some name large workflows, uh, and named wildcards are an efficient way of making sure that your workflow is very generalized, and you can switch the workflow between projects and share it between different people. Okay. Uh, so I hope everyone is able to follow and has generalized their rule by using named wildcards and have generated b.upper.txt. Actually, we haven't generated the b.upper.txt because there's the dry run flag over there. So I'm going to remove the dry run flag and actually run the Snakemake workflow on b.upper.txt. And there is my b.upper.txt that has been created. Okay, great. So we have finished the first part of our workflow where we have now created new files with the text converted to uppercase. Now the next job in the workflow is to concatenate the output of both these files into one single file. But if you see yours in our the rule that we have written previously, there was just one input and there was just one output for this particular rule. But for the next step, which is where we want to concatenate the outputs from both these files, now you realize that there are two inputs instead of one. And in Snakemake rule syntax, there are two ways of representing how we refer to different indices of the input. So this is one way of representing your input, is where you actually save them as different sub elements within the input separated by a comma, or you can refer to different inputs without actually naming them, but by using the value of their indices. So here in this case, you can see that you can save two different files under input and refer to them in the shell command by their name, wherein you say input dot first file and input dot second file. This is one way of selecting subfiles. Or you can also just refer to them by the value of their indices. So here input 0th element, which would be this, and input the first element, and which would be this. So these are two ways where we can these are two ways of selecting input files for your shell command. So now let's write our second rule. So in our previous rule, we have seen, like, we have generalized the rule so that it replaces the wildcards with whatever target file that we have asked. But wouldn't it be nice for a workflow could use any files, not just a or b.txt? We can achieve this using the named wildcards method that we have discussed earlier. So by using named wildcards, Snakemake looks at the rules it has available and tries to assign values to all wildcards so that the targeted files can be generated. We therefore have to name the wildcards in the output file in such a way that it contains the information about which input files it should be based on. So think about it. What we are saying here is that you should name your output files in such a way that your input files are based on it. Might not be too intuitive at the beginning, but you will get to see when we actually run the rule. I am going to paste the second rule, concatenate files, over here. And as you can see, now let's read through this rule over here. You can see that because it has two input files, we are referring it to as first.upper.txt and second.upper.txt. And the output that we want to be made is first underscore second.txt. And the shell command to actually concatenate the output of any two files is cat first file, second file, and send it to the output file. So that is what we see over here. Here we are referring to the input file by their indices. So cat input 0th element, which would be first.upper.txt, and input the second element, which would be second.upper.txt, cat these two and send it to the output file. So in this way, and you see how the wildcards have been reused in input and output, and also notice the pattern of how they have been used, because that is how we can now control, because of naming it that way, we can now control which input files to use by the name of the file we ask Snakemake to generate. And what do I do that is now, just to make sure that I've saved my snake file by pressing Control S. So I'm going to ask Snakemake to make a_b.txt, because if you remember, the output of the rule concatenate files is in the same format, first_b.txt, first_second. So now when I ask Snakemake to make a_b.txt with capital with a -n flag just for a dry run, there you can see that it has recognized the wildcards A and B as first and second. And yes, it has one job to run. So now let me go ahead and run the rule and refresh your Jupyter Lab notebook. And you'll see in the output that there is an a_b.txt. And you can see that it has the output of the concatenation of the two input files. And yeah, so that is a quick look at basics of the and the basic syntax of Snakemake. We have seen how a simple Snakemake rule looks like, and we have also understood how Snakemake works by defining the target files and when executing a workflow. And we also saw how to use named wildcards for generating generic and flexible rules. Because now you realize, after you see your Snakefile is very general and can be used for any kind of input files depending on the output file, the target files that you ask Snakemake to generate. Okay, great. So now let's move on to the next part of the webinar. So that would be looking at some of the other additional features of Snakemake, because what we have seen until here, like, what we have written in our Snakefile, it could be done by bash scripting as well. Because these are actually just shell scripts that are shell commands that are being run in the workflow. So that is like not like it's not really adding to the strength of our workflow management system. So now let's take a look at some of the features where workflow management system like Snakemake really adds value compared to a more straightforward approach like bash scripting. One such feature is the possibility to visualize your workflow. Snakemake can generate two types of graphs, a rule graph and a job graph. A rule graph is the one that shows how rules are connected, how individual rules are connected in your workflow. And a job graph is the one that shows you the execution of rules with all the associated inputs and outputs, that is different samples, and depending on how many other samples that you have in your workflow. So let's go ahead and generate the rule graph for our Snakefile, this small Snakefile that we have over here. I'm going to copy paste the command from the tutorial, and you can see that here we are asking Snakemake to generate rule graph by using the -- rule graph flag, and a_b.txt is the target file that we have used in the workflow. And we are piping the output to a tool called dart, which is used to further generate a PNG. So when you execute that, and you can see in your Jupyter notebook that you can open the PNG file right in your Jupyter notebook, and you can see that here are the two rules that are involved in our workflow: convert to uppercase and concatenate files. But notice how it is a representation of only of the rules and not any of the samples that are involved. So now let's go ahead and visualize the job graph. So I am going to paste the DAG command, which will generate the job graph.png. And if you refresh your notebook, there we go. This is the job graph. The main difference here is that now each node is a job instead of a rule. As you can see that we have two samples, two files, so A and B, so that's why we have two nodes. You can see the wildcards used in each job are also displayed. Another difference is the dotted lines around the nodes. So a dotted line around the nodes is Snakemake's way of indicating that this rule doesn't need to be rerun in order to generate a underscore b.txt. We can validate this just to make sure, sanity check, is by running snakemake a underscore b.txt again. And there we go. It's Snakemake is telling you that nothing to be done. So that's what the dotted lines around the node indicate. Okay, so we have discussed before that one of the main purposes of a workflow management system is that it automatically makes sure that everything is up to date. So this is done in Snakemake by recursively checking that outputs are always newer than inputs for all the rules involved in the generation of a target files. Now try to change the contents of a.txt to some other text and save it. Now, and we'll see what Snakemake tells us when we change the contents of one of these files. So now I'm going to open a.txt, which says this is a.txt, and I am going to change this is snakemake.txt, and I'm going to save it now. And now let's try to run snakemake a underscore b.txt, but first let's do the dry run feature before snakemake changes anything. And I'm also gonna print the shell commands and reason for execution. Okay, so now you can see that snakemake is telling you that there are two jobs to be done: convert to uppercase and concatenate files again. So notice how that the second rule, which is dependent on the first rule, is also being said that it needs to be rerun just because the input file has been updated. So now let's go ahead and remove the -n flag and execute it. And now my a.txt should have the updated text from what you have changed earlier. Okay, so again, just as a sanity check, if you do snakemake a underscore b.txt, sorry, there is now snakemake is telling you there's nothing to be done because the output file has been processed and the timestamps haven't been updated again. So this is how Snakemake keeps track of its files in the workflow that have changed and automatically make sure that any results depending on such files are regenerated. In this case, our second rule was dependent on the first rule, so that's why the second rule also had to be redone. Okay, but what about a scenario where the rules themselves are changed? It turns out that there are multiple ways to do this, but the most straightforward way to manually specify that you want to rerun a rule is to use the -R flag. So let me show an example. Okay, so the way we are going to change or what we are talking about is we saw how Snakemake keeps track of the updates on the files, but now we are talking about what happens when we change the rule itself, right? So now let's change the concatenate files rule such that it includes not just the output of the two input files, but also the name of the input files. So that we will achieve by replacing your rule like so, where you have added the concatenating the name of the input file also to the previous command. So in this case, none of the files have changed, only we have changed how this rule works. So now let's try to see if does Snakemake still tell you, does it tell you, hey, you have changed the rule? So do you think Snakemake is expecting to be run? But no, you can see that Snakemake is not expecting to be run, as we have pointed out earlier. So now it is time to force Snakemake to perform a forced execution. And the way Snakemake, the flag that makes Snakemake to go for forced execution is the -r flag. And we are asking it to produce a_b.txt. And in this case, you can see that in the reason for execution, you can see that it is saying forced execution. So this is where the -r flag help comes in handy to print out the reason for execution. Uh, So now let's go ahead and remove the dry run feature and actually force Snakemake to rerun the second rule. And there we go, it has finished the rerun with the second rule. And now let's confirm that by opening a_b.txt. And you can see now in the output, it is first listing out which files it are being concatenated and then the output of those files. So it is important to keep in mind how Snakemake keeps tracks of files. Still, there can be situations where you don't know if any rules have changed. Maybe several people collaborate on the same workflow, but are using it on different files, for example. So Snakemake keeps track of how all files were generated especially for when you're working on big projects and collaborative projects. So a way of how we can visualize all the changes of execution within a Snakemake workflow happen is by using the -d flag. In this case, we are asking Snakemake for the target file a_b.txt, and by using the -d flag and sending the output to a summary.tab separated value. And let me show you the summary.tsv. So this is the contents of the summary.tsv. And here you can find all the information on the execution of every rule. This is the output files that are generated, the date, rule, what were the input files, what were the shell commands exactly for that were performed for execution. And give a look towards the last column. The last column, you can see currently it says there's no update on the plan, but this last column is where Snakemake, you can see if Snakemake plans to regenerate the files when it is next executed. None of the files will be regenerated because Snakemake doesn't regenerate files by default if the rule implementation changes, as we have seen earlier. From a reproducibility perspective, maybe it would be better if this was done automatically, but it would be very computationally expensive and cumbersome if you had to rerun your whole workflow every time you just fix a spelling mistake in a comment somewhere. So it's up to us to look at the summary table and rerun things as needed. You can just get a list of the files for which the rule implementation has changed by using the -r flag, by um, using this command that you can find in the tutorial. The --list code changes is a very handy feature for especially asking Snakemake Snakemake to list out all the things that have been changed within the workflow so that you can keep track of which rules have been changed. And if so, then you can force execution of all the rules that have been changed. Okay, I hope that was helpful in giving some of the nuances of Snakemake. Now, you might wonder where Snakemake keeps track of all these things. So Snakemake has a hidden folder called .snakemake that you can see over here. That is where it stores all the information about the workflow. Okay, by now, just by looking at the basic two rules that we have built, and also where we have seen when we change the contents of a file, and also where we have changed the contents of a rule, uh, these are really the basics of Snakemake. And you can build advanced workflows with only the features that we have discussed here. But of course, this is just the tip of the iceberg. And there are a lot more features when you start building out your workflow, because one of the other features is scalability of the computation. And Snakemake has features for distributing or scaling up the computation on your laptop, on any resources that you have available, and also your HPC resources that might be available on your campus. So just in a quick recap, in this section, we have learned how you can visualize your workflows using the rule graph and job graph methods. We also saw how to force Snakemake to rerun relevant parts of the workflow and list those rules that have been changed. And we have also seen how logging works and how where Snakemake is storing all the information. So this actually brings us to the end of the first part of the webinar. Uh, this is by no means, as I've iterated before, these are just the beginning basics. And a good point to start off from here is the actual Snakemake documentation, very well written documentation, and you should find enough examples to get you started with your workflow. So we will take a 2-minute break now, and we will give some time, a minute or two for people to breathe. And when we come back, we will start with the sample Snakemake workflow called for RNA sequencing analysis in the same Jupyter notebook.
Satish, before you break, there's a few good questions that we should cover.
Sure. Yeah, let's take questions.
I'm going to read these off backwards because they make sense. In talking about scaling, a question has come in that says the method you showed for concatenation would not work well if you have a large number of files. How would you recommend to do concatenation if you have, say, hundreds or even thousands of input files?
I mean, off the top of my head, I don't know if it's the best solution. One of the major advantages with Snakemake is that it's written purely in Python. So if you are familiar with Python programming language, you can use pandas data frame capabilities to merge all the input files into one. I don't know if that really answers your question, but I can think of this as one of the ways where you can handle such data. So Snakemake is completely is written in Python, so it works. You can insert Python code into your Snakemake workflow as well. So I hope you explore in that angle.
Okay. If that didn't answer your question, please submit a follow-up. But in the meantime, someone's interested if you've got any recommendations on books available on Snakemake and or Nextflow?
I don't know of any books on Snakemake currently, but there are many resources available online. And the best place to start is to just by looking at other snake files generated by other groups in GitHub. That's a very good thing, like informative thing, is to just look at how other people have written their Snakemake workflows. And there are many examples on GitHub, so that's a good place to start.
Okay, and just to reiterate that in order to launch the Jupyter Notebook, you've got to click on the notification link provided it up in the upper right corner
Mhm.
once you launch it. There had been some questions from people about how to access that.
Yeah, sorry. If you were not able to follow any part of this lesson, as I've said early in the beginning, this tutorial is self-paced, and you should be able to follow this on your own in case you were able to follow it today.
The other thing you could point out too in the read the docs is that little icon here for support.
Mhm.
Lower right corner there, the little smiley face thing, smiley chat thing, that's our in-app support. So you can always reach hopefully somebody live there. All right, I don't see any other questions. Thank you, everyone. Before you leave, let me just point out quickly that in 2 weeks, we'll have our next Focus Forum webinar featuring Noah Falgren of the Danforth Plant Science Center, who will present on plant computer vision, which you may have heard of it as it's known as Plant CV, which is a computational platform for image-based plant phenotyping. But Noah says you can use this for other organisms as well. Noah will explain some of Plant CV's built-in tools for traditional image analysis and machine learning. And he'll show you that doing image-based analysis in your own research is not only achievable, but you don't need an expensive system to do it. So to register and for more information, go to our learning or events pages. Thanks everyone for attending today's webinar. Stay healthy, stay safe. Join us back in 2 weeks. Again, we're going to take a 2-minute break, and then Sateesh will do a little bit demonstration of some different aspects of Snakeflow.
Yes, so this is not the end of the webinar. So stick back, and I'll be back in 2 minutes, and we'll go over RNA sequencing analysis.